Welcome to another deep dive, everyone. Today we're going to be looking at golden retriever muscular dystrophy.
GRMD.
GRMD, yeah. As you probably know, this is a naturally occurring model for Duchenne muscular dystrophy in humans. And so anything we learn here can have big implications for treating DMD.
Absolutely.
So we've got a research article here that dives into the specifics of what's going on in those dystrophic muscles.
Yeah.
Using some pretty advanced MRI techniques.
It does. The researchers use quantitative MRI.
Quantitative MRI?
Specifically multicomponent water T2
Okay.
and extracellular volume fraction measurements to try to get a clearer picture of the muscle pathology in GRMD.
So before we go too far down that road, can you remind us why those techniques are so important?
Yeah.
We know that T2H2O and T1 mapping have been used in neuromuscular disorders studies,
Right.
but this one seems to go a bit further.
Yeah, you're right. Well, T2H2O and T1H2O mapping are sensitive to muscle changes.
Okay.
They aren't very specific. They can pick up muscle wasting early on, but they don't give us a clear answer as to why the muscle is wasting. This study actually confirmed that in the GRMD dogs,
Okay.
they saw elevated T2H2O and T1H2O values, which was expected.
Right.
But it's like seeing a check engine light come on in your car. You know something is wrong,
Right.
but you need more information to figure out what the problem is.
Exactly. Yeah, and that's where ECV
Yeah.
or extracellular volume fraction comes in.
Exactly. ECV measures the space outside of muscle cells. The study found that ECV was abnormally high in GRMD dogs,
Okay.
even in muscles that looked normal on other scans.
Interesting.
That's a key finding because it suggests edema or fluid buildup as a consequence of inflammation.
So ECV is giving us a more specific sort of look at the muscle environment.
Yes.
Now let's talk about multi-component water T2. Can you explain that in a way that even I can understand?
Sure. Essentially, it's recognizing that water in muscle doesn't relax at just one rate when we apply a magnetic field to it during an MRI. It's more complex than that. Bi-exponential modeling is used to reveal two distinct water components, T21, which relaxes quickly, and T22, which relaxes slowly.
Okay, interesting.
Yeah.
And what did they find when they applied this to the GRMD dogs?
They found that T21 wasn't that different between GRMD and healthy dogs.
Yeah.
However, T22 was significantly elevated, along with its relative fraction called A2.
Wow.
This suggests that the slow relaxing water component is much more prominent in the dystrophic muscles.
So by looking at these different components of water relaxation, we're getting a more detailed understanding of the changes that are happening within the muscle.
Precisely. And here's where it gets really interesting. When you combine the ECV and the increased A2 findings together, they strongly suggest that the elevated T2H2O and T1 we see in GRMD dogs are largely due to edema, which, as we discussed, is linked to inflammation.
Right.
So it helps us to explain the underlying cause of the abnormalities we're seeing on the less specific MRI measures.
That makes sense. So we're moving from a general observation of muscle changes to a more specific understanding of the actual mechanisms at play.
Yes.
What else did this study reveal about the disease process in GRMD?
Well, they also looked at how these markers changed over time, from 2 months of age to 49 months of age.
Oh, wow. So over years.
Yeah, and they found that T2H2O, T1, ECV, and A2 actually decreased with age in both healthy and GRMD dogs.
That seems counterintuitive.
It does, doesn't it?
Why would those values decrease in dogs with a progressive muscle disease?
It's actually reflecting normal muscle growth and maturation.
Oh,
As muscles develop, the cells get bigger, they contain more protein,
right.
and this affects how water behaves within the tissue.
Okay.
However, the key finding is that the decrease in these markers was much faster in the GRMD dogs.
So while both groups experienced these changes related to just muscle development,
Right.
there's something else happening in the GRMD dogs that's causing these markers to decline more rapidly.
That's the burning question, isn't it?
It is, and it leads us to one of the most important aspects of this study.
Exactly. The faster decline we see in those markers in GRMD dogs is likely connected to the inflammation subsiding as they age. You see, chronic inflammation is a key characteristic of GRMD, especially in the early stages. As that inflammation lessens, so does that edema, which leads to a quicker reduction in ECV and A2.
So we're essentially seeing the disease process play out in those marker changes over time. That's got to be incredibly valuable for research, right?
Absolutely. One of the biggest hurdles in neuromuscular disorder research has been the lack of really sensitive and specific biomarkers. We need reliable ways to track how the disease is progressing, how well treatments are working, and to identify which patients might respond best to which therapies.
And these new techniques, using ECV and multicomponent water T2 analysis, could provide those biomarkers we desperately need.
They have the potential to revolutionize how clinical trials are designed and how we measure treatment success.
It's exciting to think we might one day be able to precisely measure how a new drug is impacting inflammation and edema in the muscles. Are there other ways these techniques could be applied?
Definitely. Multi-component water T2 analysis is already a mainstay in brain research, think myelin water fraction. And if it becomes as widely adopted in muscle research, it could open up entirely new avenues for developing more targeted and effective treatments for these debilitating muscle diseases.
So not just better diagnosis, but potentially treatments that address the very mechanisms causing the disease in the first place. This research is really eye-opening. It feels like we've only just begun to explore what these advanced imaging techniques can do.
There's so much more to uncover about the intricacies of muscle pathophysiology, and these sophisticated MRI techniques are giving us the tools to do just that. We're at a really exciting point in this field, on the verge of a new era of understanding and treatment for these devastating diseases.
The study also emphasized the importance of the longitudinal aspect. They followed these dogs over a significant period of time. What did we learn from that long-term perspective?
Following the dogs over time highlighted just how dynamic the disease process is and how much it changes as the dogs age. We saw that even healthy dogs experience changes in those markers as they grow. This tells us we have to be very careful when we're interpreting MRI findings, especially in young, growing animals.
So normal development has a significant impact, and we can't assume that every abnormality we see on an MRI is solely due to the disease.
That's a key point. It also has major implications for clinical trials. We need to make sure we're using age-matched control groups and establish those baseline values for the markers at different developmental stages.
That makes sense. Otherwise, we might misinterpret the results and not get a true picture of whether a treatment is working. Let's delve a bit deeper into the specifics of the multi-component water T2 analysis they used. They used a bi-exponential model to look at those two components, T21 and T22.
And you'll recall that it was T22, the slow relaxing component, that was significantly elevated in the GRMD dogs compared to the healthy dogs.
Right, and the researchers suggested that this T22 component likely reflects the water in the extracellular space, which ties directly back to ECV being a measure of that space.
Exactly. Both ECV and A2, the relative fraction of that T22 component, were elevated in the GRMD dogs. This pointed to an expansion of the extracellular compartment, which they attributed to edema, that hallmark of inflammation we've been talking about.
So we're building a more precise picture here. The traditional T2H2O and T1 measures provide a broad signal that something is off. But ECV and this multi-component T2 analysis let us zoom in on the source of that signal with much greater detail.
It's like using a high-resolution microscope instead of a blurry magnifying glass. That level of detail is essential if we want to develop treatments that target the root cause of the disease, rather than just treating the symptoms.
One finding that stood out was that the decrease in ECV and A2 with age was much faster in the GRMD dogs compared to the healthy controls.
We talked about how this is likely tied to the inflammation subsiding as the GRMD dogs age. Remember, chronic inflammation is a key feature of this disease, especially early on.
But here's a question that popped into my mind. Wouldn't that chronic inflammation eventually lead to fibrosis or scarring in the muscle tissue? And wouldn't fibrosis also show up in the ECV signal?
That's an excellent point, and it highlights one of the limitations of ECV on its own. While ECV is very sensitive to the expansion of the extracellular space, it doesn't tell us what's causing that expansion.
So both edema, the fluid buildup, and fibrosis, the scarring, could contribute to an elevated ECV reading.
Precisely. And that's where this multi-component T2 analysis really shines. Studies have shown that fibrosis doesn't have a big impact on how water relaxes during the MRI, that T2 relaxation we've been talking about. So if we see A2, the fraction of that slow relaxing component, decreasing, it's less likely due to fibrosis and more likely a genuine decrease in edema.
That's a crucial distinction. So early on in GRMD, both ECV and A2 might be high because of inflammation and edema. But as the disease progresses, that A2 measure could give us a clearer picture of how the edema is responding, either to treatment or just the natural course of the disease.
Exactly. Think of it like having two gauges on your car's dashboard. One might tell you the engine temperature is high, but the other might tell you whether it's because of a coolant leak or a problem with the thermostat.
And having both readings gives us a much better understanding of what's actually happening under the hood. This emphasizes that key takeaway from the research, the power of combining ECV and multicomponent T2 analysis. It's about using both techniques together, not just one or the other.
They complement each other beautifully, like two pieces of a puzzle that, when fitted together, reveal a much more complete and detailed picture of what's going on in the muscle tissue.
This reminds me of our earlier discussion about the potential of multicomponent T2 analysis in muscle research. You drew a comparison to myelin water fraction in brain imaging.
Yes, in brain research, multicomponent T2 analysis has become a fundamental tool for studying myelin. Myelin is that fatty substance that insulates nerve fibers and helps them transmit signals efficiently.
And what's really remarkable is that myelin water fraction, which we get from multicomponent T2 analysis, has been shown to be a very sensitive and specific marker for myelin content, even when there are other changes going on in the brain.
It's true. And that brings up a very exciting possibility. If we can achieve that same level of specificity in muscle research, using multicomponent T2 analysis to isolate signals from different compartments within the muscle tissue, it would be a huge breakthrough.
Imagine being able to differentiate between edema, inflammation, fibrosis, and even muscle fiber degeneration, all non-invasively, just using MRI.
The potential is truly remarkable. It underscores how important it is to invest in research that continues pushing the boundaries of these advanced imaging techniques.
Before we move on, I wanted to revisit something we touched on earlier. There was a discrepancy between ECV and A2 in the study, with ECV consistently higher than A2 across all the age ranges they looked at.
Right, and we talked about how this difference is probably related to ECV being sensitive to both edema and fibrosis, while A2 is mainly driven by edema.
Since fibrosis tends to accumulate over time, that could explain why the difference between ECV and A2 gets bigger as the dogs get older. Do you think this difference between ECV and A2 could actually be a useful indirect marker for fibrosis in the muscle tissue?
That's the burning question. If we could use this non-invasive imaging technique to assess fibrosis, it would be a game changer, especially in conditions like Duchenne muscular dystrophy, where fibrosis plays a major role in muscle dysfunction and loss of mobility.
It would give clinicians a powerful tool to monitor how the disease is progressing and make more informed treatment decisions.
It could even open the door to developing therapies that specifically target fibrosis, potentially slowing down or even reversing the course of the disease. It's amazing to think that such subtle differences in MRI signals could have such a profound impact on how we understand and treat muscle diseases. This deep dive has really highlighted how groundbreaking this research is.
Absolutely. And as we continue to unravel the complexities of muscle biology, I have no doubt we'll uncover even more fascinating discoveries that will lead to better treatments and hopefully cures for these debilitating diseases.
Well said. I think that brings us to a natural point to wrap up this deep dive. It's been a pleasure discussing these exciting advances with you and our listeners.
The pleasure has been all mine. As always, we encourage everyone to keep learning, keep asking questions, and keep exploring the frontiers of scientific knowledge.
Because it's through that spirit of curiosity and inquiry that we unlock the secrets of the universe and pave the way for a brighter future.
From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging. It is incredible to think about how these subtle differences in these MRI signals could lead to such significant advances in how we treat muscle disease. But before we get too carried away with all the potential, I think it's important to acknowledge that there are still some challenges and limitations to overcome with these techniques.
You're absolutely right. No scientific technique is perfect, and multi-component T2 analysis, especially when we're talking about muscle tissue, is no exception.
What are some of those hurdles that researchers are facing in this area?
Well, for starters, getting reliable multi-component T2 data from muscle tissue is technically demanding.
Okay.
It requires specialized equipment,
Right.
carefully calibrated protocols, and robust analysis methods to effectively separate those different water signals.
So it's not as simple as just plugging the data into a computer program and getting a clear result.
I wish it were that easy.
Yeah.
There's a lot of fine-tuning and expertise involved to make sure the measurements are accurate and can be reproduced reliably.
I imagine things like motion artifacts.
Yes.
Even slight movements during the scan and variations in muscle composition could also complicate things.
Exactly. Muscle tissue is very complex. You have different fiber types, varying amounts of fat, blood vessels. All of these things contribute to the complexity of the MRI signal.
Right.
And even the slightest movement during the scan can introduce errors that skew the results.
So there's a lot of ongoing research, I imagine, to kind of refine these techniques, develop more robust algorithms, and find ways to kind of manage these challenges better.
Absolutely. There's also a need for more standardized protocols and reference values.
Yeah, that makes sense.
This would help ensure that Results from different studies can be compared and interpreted consistently.
It makes sense. If everyone is using different methods and terminology, it's hard to have a productive conversation, let alone make meaningful progress.
Precisely. Establishing that common ground is crucial if we want to move the field forward and translate these research findings into clinical practice.
So it sounds like there's still work to be done, but we're definitely heading in the right direction.
I'm confident that we are. The future of muscle disease research is incredibly bright, and these advanced imaging techniques are giving us a glimpse into a world we couldn't see before.
It's like we're finally getting a backstage pass to see the inner workings of the muscle, that complex dance of cells, molecules, and fluids that allows our muscles to function.
That's a beautiful way to put it. With each new insight, we move closer to understanding the root causes of these diseases and developing therapies that can truly make a difference.
This has been a truly fascinating deep dive exploring some cutting-edge research.
The pleasure was all mine. It's been a great discussion.
From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.